So that's B cell maturation. Questions about that before I kind of move on to now T cells, T cell receptor, and T cell maturation. So I'm going to go, like what I just did was B cell maturation. Basically, what do B cells do as they go through the maturation of the bone marrow, but before they encounter antigen is mostly what I talked about so far. Does that make sense, everybody? So we're going a little bit of a time course here. Now we're going to do the same thing for T cells. How do they mature and then get released to circulate before they encounter antigen? We're going to go through the same kind of general process here, but now for T cells. And that's going to lead into then what happens during activation of these guys. Okay. So T cell receptor, T cell maturation is then kind of now what we're going to go through. So T lymphocytes, these are first made during hematopoiesis in the bone marrow. But then immature T cells are going to basically migrate from the bone marrow through the circulatory system to the thymus gland. And they're called T cells because they mature in the thymus gland. They go through a maturation process in the thymus gland and a training process in the thymus gland that we're going to talk about. And most of the stuff that I'm going to discuss today and a little bit tomorrow is T cells in the thymus gland, most of it. T cells, though, are going to express a unique antigen binding molecule called the T cell receptor. Again, if B cells, they make antibodies. That's their antigen binding molecule. But we call it antibodies because sometimes the antibodies are membrane bound as part of the B cell receptor, but sometimes they're secreted out and they're soluble. So that's why we call the antibodies one term. T cell receptors are always membrane bound. They have a lot of similar structural features, though, to an antibody. They're probably related to one another. And then there's two different types of T cells. There's actually way more than two different types of T cells, but there's two main subtypes, maybe a third subtype of T cells. But the two main subtypes that we're going to talk a little bit about are T helper cells versus T cytotoxic cells. So TH cells versus TC cells. Okay. T helper cells. We already talked a little bit about these guys, but it's good to go through a little bit of review. T helper cells have a T cell receptor and a CD4 co-receptor. So they always, T helper cells have both the T cell receptor, but also a CD4 co-receptor. And those are expressed together. And the function of a T helper cell is they are the major cytokine producer of our adaptive immune system. And they're really needed for a strong immune response. By making their cytokines, when they're activated, they activate a lot of other immune cells. So that the main function of T helper cells is cytokine production and activation of strong immune responses. Versus a T cytotoxic cell, it has a T cell receptor with a CD8 co-receptor complex. And its job is cell killing. So it has what are called cytotoxic, and that just means cell toxic is what that stands for. Cell killing activity. But really, it's going to be, its main job is killing host cells that have intracellular pathogens or that are cancerous. So we're going to talk a little bit about these guys several different times. Now, again, this is a little bit of review, but it's nice setup for remembering how these different types of T cells function. But we're going to take a closer look at that T cell receptor, how it's made, how it functions in either T helper cells or T cytotoxic cells. The structure of the T cell receptor hasn't actually been known for nearly as long as antibodies. Antibodies had these nice soluble forms floating around in our bloodstream, were easy to harvest. and easier to study. It wasn't until the mid to late 80s that we started to actually know what the T cell receptor structure was. This because the T cell receptor is always membrane bound, which makes it harder to study. The other big problem is the T cell receptor does not sense free antigen. So the B cell receptor in antibodies, it can just detect antigens that's kind of free floating all by itself. Remember, the T cell receptor doesn't sense free antigen. It only senses antigen that's been complexed to an MHC, major histocompatibility molecule. So a lot of times you have a target cell or antigen presenting cell, MHC, T cell receptor, and basically all the signaling complex is sandwiched between two cells. So if it's hard to study a membrane-bound protein, it's even harder to study two membrane-bound proteins that are sandwiched between two cells, in essence. So it was, it's never secreted, which made it harder to study. It's also much more specific than an antibody, and that made it hard to study some of the complex interactions that were going on as well. That being said, they did get this wrong. And what happens is the T cell receptor, is again specific for some type of what is called MHC molecule or major histocompatibility complex, MHC. MHCs are always involved in presenting antigens to some type of T cell. But there's two different classes of MHC. There's MHC class 1 versus 2. MHC class 2 molecules are only found on specialized antigen presenting cells. And their job is to present antigen specifically to T helper cells. So MHC class 2 is to T helper cells. And so a combination of antigen, MHC2. I didn't like that. Let's try that again. A combination of MHC class 2 and antigen is then going to present to a CD4 and a T cell receptor coming off of a T helper cell. So the CD4 and T cell receptor comes off of the T helper cell, and it's presented antigen via MHC class 2 coming off of some type of specialized antigen presenting cell. Can you guys remember any examples of antigen presenting cells? So the main one's dendritic cells. Noah, you said one, I think. Macrophages sometimes. Any others you guys can think of? Sometimes B cells, actually, can function as antigen presenting cells. And we'll talk a little bit about that as well. Okay. Now that's MHC class 2 and again, T helper cells. Those two go together. There's also that MHC class 1. MHC class 1 is expressed on the surface of all nucleated cells. And MHC1 antigen complexes, MHC class 1 presents antigen to T cell receptor CD8 co-receptor complexes on a T cytotoxic cell. So again, here's my T cytotoxic cell at the bottom, but it has a CD8 co-receptor attached to a T cell receptor. This recognizes antigen presented by MHC class 1 coming off of an antigen presenting cell, although really we kind of tend to use the nomenclature of target cell some more commonly with this. When activated, T cytotoxic cells then exhibit cell killing or cytotoxic activity. They kill off host cells that are infected with intracellular viruses, intracellular bacteria, or that are cancerous, most commonly. Okay. Questions about that? The setup here. Because now we're really going to start taking a look at the T cell receptor more specifically that is involved. And tomorrow, we're going to start talking about major histocompatibility complexes in more detail. MHC class 1 versus MHC class 2, but also antigen presentation is going to be what we're talking about a little bit tomorrow as well, start to talk about, and through into Thursday. Okay. All right. T cell receptor. T helper cells express CD4 or again, remember T helper cells express CD4 but not CD8. C cytotoxic cells express CD8 but not CD4. In either case, these CD4, CD8, this is basically CD4 structure looks a little bit like this. CD8 structure looks a little bit like this. Okay, but they are associated with the T cell receptor. You have either a CD4 T cell receptor complex on T helper cells, or you have a CD8 T cell receptor complex on T cytotoxic cells. So here's my T helper cell, here's my T cytotoxic cell. These function as co-receptors. Or, and in essence, they're what are called adhesion molecules in this particular case. CD4 helps with the recognition of class 2 MHC proteins. CD8 will bind to and help with the recognition of class 1 MHC molecules. And I've been showing this in the diagrams, even if you guys kind of didn't notice it. So if I kind of back up a slide here. Oops, I'll come back to this. Again, what you see in some of these different diagrams, whoops, nope, not these, is that there's this little part of, say, CD4 that's contacting MHC class 2 here. There's a little part of then CD8 that's contacting MHC class 1 here. All right, and so they're involved in those recognitions. And if I'm showing that again, that's just what's you're kind of seeing some of those things in the diagrams, but it's a little more prevalent in this space fill model. So what this is this is T cell receptor and CD4 coming off of a T helper cell or a T cytotoxic cell. T helper cell, right? And so what ends up happening is a T cell receptor, by the way, this squiggly line right here is the antigen. See the greenish right here? This is the antigen right here. And so the T cell receptor really is mostly recognizing the antigen and a little bit of the MHC class 2 complex. It's binding onto that combination right there. But the CD4 co-receptor comes in and often binds onto this part of MHC class 2 as well. And that helps with the recognition and the proper binding signals. Okay, so the binding of the T cell receptor is greatly enhanced and augmented, helped along by either CD4 or CD8 binding as well. Questions about that? Okay, so I said there's mostly T helper cells versus T cytotoxic cells, except there's not. T helper versus T cytotoxic cells are probably the two most important kinds of T cells, but there's other kinds of T cells as well. We're going to fill in some of the details there, but it turns out there's actually two different flavors or kinds of T cell receptors. There's what is called alpha beta T cell receptors or gamma delta T cell receptors. And there's some different stuff going on here. This is an immunoglobulin gene. This is antibodies from a B cell, just a structural diagram. Again, we have heavy chains, light chains. The T cell receptor has some similarities. You can see that it has these immunoglobulin folds, these immunoglobulin-like domains. It has disulfide bonds down here. Does that make sense, everybody? We're going to talk more about that here in a second. This is an alpha beta T cell receptor. This is one variety of T cell receptor. It turns out there's also a gamma delta. So Either way, though, whether or not we're talking about an alpha beta T cell receptor or a gamma delta T cell receptor, they're very similar in structure to antibody immunoglobulin genes. And if you look at it, they've got a variable domain and constant domains. So each chain, in this case, we have an alpha chain protein and a beta chain protein. Each chain has a variable domain region and a constant domain region. And they also have a little bit of a transmembrane domain region. Hydrophobic alpha helix. Okay. The variable region, variable domains of either alpha beta or also the gamma delta T cell receptors are very similar. They come together, variable alpha, variable beta, to make the antigen binding cleft. So it's this part, the variable domain, that recognizes antigens. And it's very, very similar to the variable domains from antibodies that we just talked about. In fact, pretty identical, but we'll get to that here in a second. And so those are the antigen binding sites. But again, sometimes you have an alpha beta T cell receptor, sometimes you have gamma delta T cell receptors. Most T cells express the alpha beta variant of a T cell receptor. So 95% of the time, you get a T cell with an alpha beta T cell receptor. About 5 to 10% of the time, you get a gamma delta T cell. Gamma delta T cells, if you have a T cell that has a gamma delta T cell receptor, these T cells tend to be abundant along epithelial surfaces. So the epidermis, they tend to be abundant along intestinal surfaces and urogenital tract surfaces. So they tend to be particularly abundant along mucosal surfaces. And they have pretty similar structure. Here's an alpha beta T cell receptor in terms of its structure. But it has, between the variable domains and the constant domains, it has a bend of 147 degrees. So that's its shape. Whereas the gamma delta T cell receptor has a bend of 111 degrees. So it has a slightly different shape. But remember, shape correlates to function. And so as a result of their changed shape, they have slightly different functions. So remember, about 5% of cells are gamma delta T cells, and it turns out gamma delta T cells are not MHC restricted. Gamma delta T cells, if they have a gamma delta T cell receptor, they actually bind free antigen. Similarly to what antibodies and B cell receptors do. So again, an alpha beta T cell receptor needs antigen presented by MHC. That is called MHC restriction. If T cells have the gamma delta variety of a T cell receptor, they can detect free antigen that is not complex with MHC. But these T cells are not very common in our body. They're at a fairly low level. Okay. And the idea is these gamma delta T cells may be actually our first lines of defense when it comes to adaptive immunity. They may function a little faster than the alpha beta variety of T cells. And so they can actually function first and limit the infection until then an MHC restricted alpha beta T cell response is mounted. So these are kind of our first responder version of a T cell. Okay. But again, only 5% of our T lymphocytes are the gamma delta variety, have that gamma delta T cell receptor. Questions about that? The T cell receptor, again, looks a little bit like this. Do you guys see these little tiny short cytoplasmic tails? Remember how that was a problem with B cell receptors? It's also a problem with T cell receptors, whether or not it's alpha, beta, or gamma delta. Turns out that T cell receptors are also going to complex with what is called a CD3 co-receptor. So T cell receptors always are expressed in conjunction with CD3. And CD3 is a group of proteins that's just collectively referred to as CD3, and it forms a co-receptor. And the T cell receptor and the CD3 co-receptor proteins are always co-expressed together. And so if you have CD3, you typically also have a T cell receptor. If you got both of those, you got a T cell. CD3 is actually a complex of 5 different polypeptides that interact with the T cell receptor, and it's a gamma, delta, epsilon, zeta, and eta protein. I told you guys you were going to get sick of the Greek letters, and so I apologize. I didn't name it though. Okay. Typically, the CD3 complex is a copy of gamma and epsilon that form a heterodimer. Then a second copy of epsilon and delta form a second heterodimer. And then you get, and so that's these guys. So here's my alpha beta T cell receptor. This will complex with an epsilon and gamma protein, and then an epsilon and delta protein complex. And then in addition to all of that, you also either get zeta or eta. In this case, I have a zeta-zeta homodimer, but sometimes you get an eta homodimer as well. Zeta and eta are actually encoded by the exact same gene, but the two forms are generated by a differential RNA splicing. And about 90% of the time you get zeta-zeta, and about 10% of the time you get eta-eta. But usually the CD3 complex consists of zeta, zeta, gamma, epsilon, epsilon, delta. Questions about that? And those are a co-receptor complex that attach onto the T cell receptor. And that's for both the alpha beta T cell receptor, but also the gamma delta T cell receptor. Okay. With the T cell receptor, the short cytoplasmic tails of alpha beta or gamma delta chains suggest they don't really function to really transduce a signal fully across the membrane. They need the help of the CD3 co-receptor complex. That CD3 co-receptor complex has longer cytoplasmic tails that are involved in signal transduction. And if you mutate either CD3 or the T cell receptor itself, you lose signal transduction. Both of them are necessary for proper function. Transmembrane domains of all the CD3 proteins contain a negatively charged aspartic acid in the middle of the membrane, whereas the alpha beta and gamma delta things, they have positively charged lysine and arginine residues. So you have in the transmembrane domain of the T cell receptor, you have some positively charged amino acids. In the transmembrane domains of the CD3 co-receptors, you have some negatively charged interactions. It turns out the ionic interactions help to hold the complex together. So the T cell receptor is going to recognize the antigen MHC complex. And then the CD3 co-receptor will actually then transmit the activating signal into the T cell itself. And that's going to involve phosphorylation of ITAM domains. So I said with those, we're going to come back up at one point. But you have several ITAM domains on gamma, epsilon, epsilon delta, and then zeta, zeta, or eta, eta. So these are all the ITAM domains in blue. But the ITAM domains, these are phosphorylation or kinase domains. So phosphorylation occurs at these ITAM domains, immunoreceptor tyrosine-based activation motifs, and that results in a phosphorylation signaling cascade that turns on T cell responses. And this is very similar to when we talked about the B cell receptor as well. Okay. All right, last thing I want to talk about today, and this is kind of, we're going to go in through, this is kind of set up for then tomorrow. We're going to go through it in more detail, is then organization of then the T cell receptor genes. And again, this is something I find that's kind of good. This is a good breaking point. We go through this, introduce it, and then we'll review it tomorrow as well. So organization, expression of the T cell receptor genes. All right, the genes encoding the T cell receptor. Alpha and delta genes are co-localized on human chromosome number 14. So again, alpha chain genes are on 14, so are delta chains, and they're actually mixed in with one another. Same spot on chromosome number 14. Beta chain and gamma chain are also co-localized, are on chromosome number 7, but interestingly enough, different parts of chromosome number 7. So one's over here and one's all the way over here, and they're not intermixed with one another. So these alpha and delta are intermixed. But why am I emphasizing that? It's actually going to become important when we talk about gene rearrangements. Functional T cell receptors are also going to require DNA recombination, DNA rearrangements, the same way that antibody genes are created. Okay, so everything we just finished talking about for immunoglobulin gene rearrangements is going to come right back up for T cell receptor gene rearrangements, but with just a few small differences here and there. Okay. So this is the organization of genes encoding T cell receptors. They are multi-gene families that have, again, constant gene segments, and then constant regions that code for the constant domain, but also a series of gene segments that are rearranged by DNA recombination to make a variable domain. Alpha and gamma variable domain is composed of V and J gene segments. So here is my alpha chain rearrangement, but you got to be careful. Remember, alpha and delta are co-localized on the chromosome 14 in humans. So what you have is variable alpha, variable alpha, variable alpha. You have all of the variable alpha gene segments here. Then what do you have? You get all of the variable delta gene segments here. Then you've got the delta D segments and the delta J segments here. Does that make sense? And then you got the delta constant segment. Okay. Now you can flip over here and you've got the J segments for alpha and the constant segment for alpha. What that means is you've got alpha on one side, alpha on the other. So this is going to become important. Alpha, alpha. Okay. So this is the alpha, and this is the other parts of the alpha. And in the middle, you've got deltas, all of the delta. Everybody okay with that? Alpha has V segments and J segments, but no D segments. So is it similar to light chain or heavy chain? Alpha is similar to light chain. Gamma is then down here. It also has V and J segments, but no D segments. So gamma is also similar to light chain. Beta and delta have V, D, and J segments, so they're similar to heavy chain. Everybody okay with that? Okay. Now, the other thing I pointed out is the presence of these delta genes here is significant. Why is it significant? The delta gene location is significant because if alpha chain rearranges, then it deletes delta. Remember, when you do a gene rearrangement, it deletes any sequence in the middle. So if you have an alpha arrangement, say alpha V segment A rearranges with J segment, all of that intervening sequence, which is the delta sequence, gets deleted permanently out of the T cell. What that means is you can have alpha or delta, but never both. You can never have alpha chain and delta chain co-expressed in the same T cell. Successful rearrangement expression of alpha deletes out delta. And that sets up T cells, again, can only be an alpha beta T cell receptor or a gamma delta. Gamma delta T cell receptor. Okay. It never both is what that basically ends up being. Okay. So again, if you recombine here and here, you delete all of delta in the middle. Questions about that? Gamma. Eventually, yeah, there, I remembered how to write it. Okay. Okay. When it comes to the rearrangement, all of the mechanisms for rearranging these genes are very similar to what happened with antibody genes, immunoglobulin genes. You're still going to have 1-turn and 2-turn recombination signal sequences. You're still going to be using RAG1 and RAG2 recombinase enzymes. So all that stuff that was involved with B cell arrangements in the bone marrow is now being used for T cell rearrangements, T cell receptor rearrangements, but now in the thymus gland for the T cells. So you're still going to have upstream and downstream of these variable segments or J segments or D segments. You're going to have 1-turn recombination signal sequences or 2-turn recombination signal sequences. You're going to have RAG1, RAG2 enzymes binding to those and mediating, cutting the DNA and stitching together a combination of the DNA. So, and it's the same rules. Say you're doing gamma. Remember, gamma has V and J segments. So you join together by DNA rearrangement V and J segments. Alpha, you join V and J segments. For beta and delta, if you use those, you do DJ joining first, and then V to DJ joining. So it's the same order as in heavy chain rearrangements. Okay, just some slightly different details. That's what I mean by sometimes this next coming exam on Tuesday is easier, so yet sometimes also harder because we got the same thing twice with some slight variations. Okay, questions about that? And this diagram kind of shows it. You're doing, this shows alpha chain rearrangement steps versus beta chain rearrangement steps to get an alpha beta T cell receptor. With the alpha rearrangements, if alpha rearranges, again, you bring a variable alpha together with a J alpha segment. This is then the rearranged DNA right here, but then you go through RNA splicing to then get to the final expressed alpha chain. Same thing here with beta, but again, you have V to DJ joining. You have DJ joining first, and then you have, DJ joining first, and then V to DJ joining. Questions about that? All right, we're going to stop there, and I'm going to pick back up with this. But that is, again, the recombinatorial diversity that gives rise to T cell receptor diversity. But there's also going to be combinatorial diversity and junctional diversity to make different kinds of T cells as well. But we'll pick back up with that tomorrow.